Nat 5 Biology, Cell Biology video number 3, Genetic Engineering and Respiration. If we firstly have a look at genetic engineering now. DNA can be transferred from one cell to another, either naturally or artificially. Genetic engineering involves artificially transferring sections of DNA from one organism to another, and the resulting organism is said to be genetically modified. It will now use the new genetic material in exactly the same way as its own and manufacture any protein that new genetic material codes for. Now, bacteria are very useful in genetic engineering. They have the main DNA in the form of a loop. They also have smaller circles of DNA called plasmids. These plasmids can be removed and used in genetic engineering. If we take one of these plasmids, extracted from the cell, we now need a gene to insert into that plasmid. In this case, it's a gene taken from a human cell. The required gene is identified and cut out using enzymes. That required gene is then placed into the plasmid. The plasmid's opened up using enzymes, and different enzymes enable for that gene to be inserted into the plasmid. The plasmid's now taken, put inside another bacterial cell, usually of a different species, and then that bacterium is then taken to some kind of medium, like inside a petri dish, some agar maybe, in order for that cell to grow. Under the correct conditions, the cell will multiply until there are millions of them, each one containing the required gene that's been inserted into the plasmid, because all the genetic material is copied within the bacterial cell when that cell divides. So all of these bacterial cells will have the potential for producing whatever protein it is that is coded for by that required gene inserted into the plasmid. Here are some examples of products made by genetically modified bacteria: insulin, interferon, and human growth hormone. If we now look at the process of respiration within cells, respiration involves the breakdown of glucose. All cells require a source of energy, and glucose is the main source of energy in living cells. It's an energy-rich substance, a type of sugar. It's the most common product at the end of digestion of complex carbohydrates like starch, and the chemical energy stored in glucose must be released by all cells through a series of enzyme-controlled reactions called respiration. Now, energy is transferred within a cell by way of a substance known as ATP. If we consider glucose and oxygen, the raw materials required for respiration to occur, the energy from the breakdown of the glucose through respiration is used to generate ATP. That ATP can then be used in other parts of the cell to carry out certain functions, different jobs, such as the synthesis of proteins from amino acids. So the ATP that is produced from the release of energy from respiration can be used in energy-consuming reactions such as muscular contraction, cell division, protein synthesis, transmission of nerve impulses, the active uptake of ions and molecules by active transport. Now we look at the first stage in respiration, which is called glycolysis, and takes place in the cytoplasm of cells and does not require oxygen. The glucose is broken down into 2 molecules of pyruvate, and in that process 2 molecules of ATP are generated. Now, the second stage of respiration depends on whether oxygen is present or not. If oxygen is present, then respiration continues along the aerobic pathway, and that occurs in the mitochondria. If there's no oxygen present, then the fermentation pathway takes place, and the process is only carried out in the cytoplasm. So if we now look at the aerobic pathway, this takes place in the mitochondria. The mitochondrion is a double membraned organelle within the cytoplasm of a cell. The inner membrane is highly folded to provide a very large surface area for all the enzymes that are required to carry out the second stage of respiration. So if we summarize that second stage and the process of respiration as a whole, like we've already seen, glucose is broken down into 2 molecules of pyruvate. that is called glycolysis and takes place in the cytoplasm. That is the first stage. The second aerobic stage of respiration occurs in the mitochondria, and here each molecule of pyruvate is broken down to produce water and carbon dioxide. And in this second stage, this second aerobic stage, many molecules of ATP are generated. The aerobic pathway of respiration is by far the most efficient at releasing energy from the breakdown of glucose. So if we now look at respiration when there is no oxygen present, this is called fermentation, and occurs in animal cells during vigorous exercise, for example where muscles do not receive enough oxygen. Those cells undergo fermentation and they produce a product called lactate, and this causes muscle fatigue or cramp. The glucose is first converted to pyruvate, but then lactate is generated from pyruvate and only a little bit of energy. To be precise, that's only 2 molecules of ATP, as we saw in glycolysis. So, the lactate that is produced from the lack of oxygen, that lactate can be converted back into pyruvate once the exercise is over and the muscles can receive more oxygen again. Now, this conversion of lactate back into pyruvate, which then goes on to complete the aerobic pathway of respiration, producing lots of ATP, that is called repaying the oxygen debt that was built up initially from the lack of oxygen, which generated the lactate in the first place. And finally, fermentation in plant cells and yeast cells. Fermentation can occur in the root cells of plants in waterlogged soil or in yeast cells deprived of oxygen, such as when we make beer or wine. And when the plant and yeast cells respire anaerobically or without oxygen, they produce ethanol and carbon dioxide. Here the glucose is first broken down to pyruvate, which is then broken down to produce ethanol and carbon dioxide. As the carbon is lost in carbon dioxide, the process is irreversible, as the carbon dioxide is lost to the air. So the ethanol alone cannot be converted back into pyruvate.